honoring us with your presence to be here as a keynote speaker for the 2026 annual review and work planning workshop of the Nrcri. How many of you were schooled, were thoroughly educated yesterday? I learned a lot of things about scaling. I learned a lot of things about how to move agriculture to the transformative mode from his talk. And he's one of those people that engages you in his discussion. I know many times our keynote addresses are being given, and I would doze off and doze back. But yesterday, he was like making you hungry for knowledge. So thank you very much, sir, for yesterday's, it was a classical session, a classical keynote address that we had that's a model. And the things we were talking about, you were speaking to Nigeria, you were talking to us. And you were also talking to, let's say, Africa, because we are being live streamed both on Facebook and YouTube, and I'm getting many feedback from all over the world about what we are doing. So many people are listening to you. Many will still listen because it's still right there, saved in our YouTube page. So thank you for coming all the way from Addis Ababa, and we feel blessed, not just because you came to give a lecture, but because we have started a new journey, a new relationship, and an open door, or let me be more broad, many open doors in one person. And so join me to give him a rounded clap of applaud as we give this.
Thank you, Madam Chair. So thank you very much to the Executive Director, Prof. Chiedozie Egesi. And congratulations to all our awardees. So we'll continue. Our next stop is the potato program. Potato program. Yeah? Just potato, not sweet potato, potato program. Yeah, because the sweet potato is much later. Potato program. Thank you.
Thank you, Chairman. Scientists, please permit me to stand on the already established protocol. I want to start by saying that I am grateful to be here with our strong supporters from Sepia Foundation and from Sahel Consulting. Thank you for coming. I want to start the presentation by showing the key achievements of the program for 2025. As you've all heard through our platform in the Institute and other sources, we have registered a potato variety that is called Connect. It was registered last week, and it is a great achievement for the program and the Institute. Yeah, we are also conducting more evaluations for 3 more new varieties that, by the grace of God, will soon be registered and released also. We conducted on-farm evaluations and multi-location for 3 different hybrid true potato seeds, not seed tubers. Okay, not seed tubers, but hybrid true potato seeds. It is still undergoing evaluations. We have had our first year and on station. So as the season comes, we will continue with more evaluations. And in the past year, we had a lot of collaborations with Sahara Consultants, Sepia Foundation, Solinta Potato Company, Fruits and Veggies, including the local partners such as Plateau State ADP and the Plateau State government as a whole. Next slide. Next slide. Yeah, I want us, no, please go back to the pictorial slide. I want to show us this particular slide to enable us understand better what we have been talking about. The Connect Potato we released. The field evaluations is the first one that we have there by the left-hand side. You can see from other potatoes near to that particular place where I am standing, Connect is showing a very good potential for performance in Nigeria. The second picture there is the true potato seeds that seedlings in trays inside the screen house. And the one next to it is where we are conducting some trials with spray tube irrigation. We are using spray tube because the stems of the TPS are so tender that when strong droplets from a normal sprinkler irrigation drops on them, it breaks them or destroys them. So we now adopted this method of spray tube irrigation. The other pictures there by the right-hand side is the true potato seed cans that were obtained from the Solinta company. The cans are there, and the one on red is the seeds that are coated. And below it, the first one is the potatoes harvested from the Connect, and the ones in the car are the ones we harvested from the 3 varieties that are going on. The next to the last pictures there is the program exhibition stand at the potato fair. If you look at the last one, you can see our executive director also attended the fair. He is there with the program, standing at our stand where we were exhibiting the materials that we had from the program. Next slide. Next slide. Yeah, this is the first work here is evaluation of varieties for adaptation and performance across potato producing areas of Nigeria. Here we are evaluating 3 different varieties, 3 different lines, including Arizona, Paradiso, and Alouette. And we are using 2 checks against them. One of them is Nicola, which we are using as the local check, while Unica, we are using it as the hybrid check. Unica is one of the materials that was released in the last release in 2023. And from the work, you can see in the table that the Arizona, Alouette, and Paradiso have high yields, more than both the hybrid check and the local check. And we discovered that these materials, based on their performance, are good for Nigerian environment and can be selected for release anytime we complete the evaluations. But for now, we still need to go for multi-location and on-farm before whatever we are going to do. Next slide. Yes, this is the Connect that we released. We want to bring the slide here to show us the qualities of the Connect. The Connect variety has a potential of yielding up to 44.2 tons per hectare. And in terms of disease, it is highly resistant to lead blight, but moderately tolerant to bacterial wilt. In terms of nutrient composition, it has a high protein and other nutrients that are required for normal nutrition. The picture under the one with green plants in the field is the tuber flesh color of the potato, Connect potato. In Nigeria, not that there's any potato that is not good, but people prefer the one that have a creamy color, both on the skin and the inside. And the middlemen have made it a little difficult for some potatoes that have a kind of skin color that is not creamy, or a kind of potato does not have a creamy color. But for Connect, it has a very good skin color, which is creamy, and also the inner flesh color is the one that is acceptable by marketers and consumers across Nigeria and elsewhere. Next slide. Yes, this picture is showing us this slide is showing the processes we passed before the release of the Connect variety. The first picture by the left there is the tubers harvested from a single stand of Connect potato. So if a single stand of Connect potato can give this, imagine what 400. The picture is only showing us that Connect is a high yielding material. Even the farmer, one of the farmers that we used during the on-farm, confirmed by showing us both the color and the tuber size that Connect is able to produce. And going towards the right, you can see the NASC technical committee. During the time they came for inspection, they were very happy. And you can see the pictures of where tubers that are harvested are packed. But the one to the last there is where we have the small tubers and everything together to show us that Connect is a variety that can produce a good number of tubers at the same time, and as well produce big tubers. So it's a good variety for Nigerians. Next slide. This one is evaluation of 2 hybrid true potato seeds in Nigeria for field performance, disease tolerance, and tuber quality. This evaluation is conducted using the true potato seed, not seed tubers, that were obtained from Solinta through Sepia Foundation with the help of Sahel Consulting. The first picture from the right there is the cans, as we showed in the very first slide that we showed you. And the seeds on the red and the pink plates are the seeds that are quoted from the company. And then the materials, once it is sown, once it is planted on the potting soil, the coating material dissolves immediately. And the germination takes just about 6 to 12 days, where the temperature is good. If you look at the picture below by the right-hand side, you can confirm that it has the potential of germinating well and sustaining its growth well. So if we are able to manage it well and do a good work, it will be a game changer for Nigerian potato production, because the most expensive stage of cultivating potato is the seed. And the seed tuber, to produce a single hectare, you need at least 26 bags of potato seed, which is about 50 to 60,000 per bag, which is going to take about 3 million naira to get the seed material to produce a single hectare. But with this technology, it will be easy. The potato true seeds are
Hello, technical crew. You need to look at the screen so that this thing doesn't go off. Can you press any button, please? Not you. I'm talking to the technical crew, please.
Okay. So the hybrid true potato seeds, they are light and can be easily transported to wherever the farmer wants with ease. The cost of the seeds is also low. The probability of transferring disease from the former generation to the new field is very well reduced. So it is going to be a very good technology if we succeed in conducting and getting the results fine to release it. It's going to be a strong game changer for potato production in Nigeria. So from the results, we see that the 012 and 017 are able to yield as much as Nicola in terms of the total number of tubers that they're able to produce. In the second, in table 2, the yield between the local check, which is Nicola, from seed tuber, is giving only 14 tons per hectare, while this one's coming from seed is giving up to 12 tons per hectare. So if you look at it, you see that the difference between planting from the seed tuber and planting from the true potato seed is not much. And if a farmer can spend less to produce a hectare and get a quantity that is almost equivalent to what a farmer can produce from the seed tubers, is going to be an excellent and profitable venture. Next slide.
Sorry, excuse me, sorry. You have just 8 minutes more for everything you need to say. And you have not given us your proposals
No
yet.
problem. The recommendations, what we recommend in this place is that the Solinta 012 that has a very good tuber size should be studied more, and that of Soli 017 that has more tuber. The two of them have to be exposed to more evaluations for further confirmations before advancing them for whatever. For the TPS, in terms of sowing them in the screen house, we have to use the recommended method of using potting soil and cocoa peat. But at the end of the day, we should not allow them to stay more than 6 weeks in the screen house before taking them to field. Please, next slide. Next slide. This is a work by one of our PhD students in this place. He is trying to study the viruses affecting potatoes in Nigeria. And from the work, he has been able to detect that potato virus A, virus M, virus S, and potato leaf virus are all present in Nigeria, and they can easily be detected by a PCR machine. And to determine the incidence and distribution of potato virus A, M, S, and potato leaf virus in the main potato growing areas of Plateau State and Taraba states of Nigeria. The next slide. What he identified in the work is that the potato virus M in a weed called Chenopodium botrytis, Capsicum annum, and Lantana camara highlights the significance of weeds as alternative hosts for virus in where potato is growing. And from his findings, he emphasized the critical role of weed management in integrated viral disease control strategies aimed to reducing inoculum sources and limiting the spread of viruses within the potato production systems. And that is the end of the reports we have. Thank you.
Go on straight to your proposals. Thank you.
Proposals.
Proposals.
Yeah.
Yeah.
In the proposals, we have first one as strengthening the traditional potato seed systems through positive selection, optimized plant populations, and low-cost technologies in Nigeria. The objective is to introduce and validate ash treatment as a low-cost alternative to chemical seed dressings, to demonstrate apical rooted cotton technology to 90 lead farmers as a pathway to clean seed multiplication, and to facilitate farmer-to-farmer diffusion of improved seed practices through field days, and to document yield improvements and cost savings for policy advocacy and scaling. Next one. The next one is field-based integrated disease management of late blight and early blight in potato production. The major objective there is to develop and evaluate field-validated integrated management strategies that combine cultural practices, biological controls, and targeted fungicide applications for simultaneous suppression of late blight and early blight diseases in potato. and to determine the economic viability of proposed integrated disease management approaches compared to conventional calendar-based fungicide programs through comprehensive cost-benefit analysis, and to assess the environmental impact reduction achieved through integrated approaches by quantifying fungicide use frequency, active ingredients, and environmental risk indicators. Next slide. This one is on farmer perception on potato viruses and their management in Plateau State. The objectives are to assess farmers' knowledge and perceptions of potato viral diseases in Jos Plateau State, and to identify the management strategies currently employed by farmers to control potato viruses, and to analyze the factors influencing farmers' adoption of virus management practices. Next one. This one is on wrap and plant technology for management of root knot nematode on potato in Jos. This technology is where a nematicide, a company produced materials and embedded them in plantain leaves. When you bury them in the ground, they can now inhibit the activities of the nematode while production of the potatoes go on. The objectives are to develop and validate wrap and plant technology using lignostic fibers, biodegradable agents for reducing root knot nematode infestation on potatoes in Jos. To assess the impact of wrap treatments on nematode population dynamics, root damage, potato yield, and quality, and to establish sustainable environmentally safe management practices that are compatible in local farming systems. Next one. This one is on farmers' perception on potato viruses. No. Please, next slide. This one is on post-harvest physiology, storage behavior, and processing quality of potatoes produced from different seed sources in Plateau State. The objectives are to characterize the initial physiological and biochemical status of potato tubers from 4 seed sources: informal farmer-saved seed, certified seed, rooted apical cotton, and true potato seed. The second one is to evaluate the effect of seed source and storage environments on the agronomic and processing qualities of potatoes, and to determine the effect of storage and on processing qualities of potatoes. This one is the G by E interaction for tuber yield, dry matter content, and specific gravity in some potato genotypes. The objective is to evaluate 10 potato genotypes for tuber yield, dry matter content, and specific gravity, and to determine the nature and magnitude of G by E interaction in potato production. Thank you for listening.
Thank you very much. Right on time. Thank you very much for speeding things up when you were talking about your proposals. So do we have any questions, please? Just in 5 minutes. Let me look this way. Whether yes. Particularly those who have not asked questions before. Sorry, okay, is that Professor Awalam Damien? Yes, okay. You'll kindly introduce yourself. So let's start with Dr. Peter, I believe. Yeah, okay, so we'll start with Dr. Peter. And then we'll come to Professor Awalam, and then we'll come this way.
Yeah, thank you very much for that presentation. My question is, what was the main practical challenge that you were having in working with the new technology of true potato seed in your station, and how do you think that that could also be a challenge for the adoption of the technology in practice?
you very much. I guess I'm known around there. Everybody knows me. At least 80% of the people, if not more than that. What I want to ask is that, Dr. Wadley, in your release of Connect, it's like you laid emphasis on the size of the tubers of the potato. I don't know if you consider the dry matter content and food quality. Remember, we've had experience in this Nrcri where crops were released and farmers rejected them because of a low dry matter content and bad food quality. I'm sure you didn't tell us anything whether you considered all those things when you were doing your research and then the release. Thank you.
Give me an asawalam, please. Behind you, behind you. Thank you.
Thank you, the chair. I actually had wanted to direct my question to the ED, but if the speaker will answer that for me, I don't mind. Most of the varieties that I've been hearing their names since yesterday are given what I would like to call exotic names. The seeds that we've been talking about yesterday, are they being produced for export or for our local farmers? And if they are for our local farmers, will they not adopt it if you give those varieties local names? That's my question. Thank you.
Thank you, Prof. So, Dr. Asamoah first.
Chris, I want to congratulate you again on the release of the Connect potato. It's an achievement. There is the first report you gave, evaluation of some potato varieties. Why don't you say evaluation of potential potato varieties? Now, the variety Connect, we are told is now resistant to lead blight. Are we now saying that we have a solution now to the lead blight that is devastating Plateau State? Are you assuring us? Remember our trip to Jos, on the assignment by the minister and the state government. Okay? What is the role of Sahel in all this? I heard the Sahel. What actually are they doing? Let's know their role. During that trip, I led the team from Nrcri. There were short and long-term solutions we developed. Are the farmers still following them to tackle the late blight? There was a project led by Dr. Amadi. I hope he's here. Since then, it's over 3 or 4 years. What is the outcome of the donor-funded project in your station? I hope Amadi is here. We are not hearing anything about it again. Thank you.
Thank you, sir. So we go behind now. I think I see Dr. Unzo. Sorry, the light is in our face. But please just give everybody raising their hands there one by one. Thank you. Yeah, you may and then keep going.
Thank you very much.
Please, let's be brief.
Dr. Wadele, I noticed that in your trial on evaluation of 2 hybrid true potato seeds, you didn't mention the initial soil properties used for the trial. Based on this, do you think you are justified to draw conclusion that the yield differences observed could be attributed solely to varietal characteristics? Because one can say, and emphatically too, that it could be as a result of differences in soil fertility trend. So I would suggest, and humbly too, that you go back and look at it, and in your future trials, try to showcase the initial soil properties. It is only when you do that that whatever conclusion you are drawing may then be sounding justifiable. Thank you.
Thank you, sir. The other hands there. Has anybody else asked the questions you wanted to ask?
Can I go ahead?
Okay, Dr. Muzo.
Yes. It's not a question as such. But I want to use this opportunity to appreciate and congratulate the AD and his team. They are doing very well. Having said that, I don't know. So that is that. Let's go with that for the time being. Appreciation of the AD and his team for the good work they are doing here. And then, especially calling ex-researchers. I don't want to say well, let me leave that. Ex-researchers. Appreciate the director also for maintaining that culture. You look at this place, everyone here has passed through here. So we want to thank him for calling us to participate, to see our old friends. I don't like using the word old. I prefer elderly people. If you talk of one being old, even the 10-year-old child is also old. So the elderly people you invite here, I want to use this opportunity, echo it to appreciate the director and his team and ask that they should continue. Praise the Lord. Praise the Lord.
Thank you, sir. So we go to Dr. Emmanuel. Dr. Emmanuel is sitting right in front, and that will be the last question for this session, please.
So, thank you, Chair. I just wanted to ask, I know you mentioned the blight, the late blight. So, Umudike is working on that with SIP. So, I didn't hear you talk about your plans. I don't know what is driving your proposals, but that is a study that is showing promising results. So, what plans do you have towards running with that? Say, for example, it is deregulated. I've not seen it there because that would be a lot of work that will involve trials, preparation, more data, on the field level. I didn't hear that. Or you are still far away from achieving that. The second thing is you post-harvest issue in seeds. What of post-harvest matters? Because there's a lot of post-harvest losses in you can't store it. So what efforts are you doing, especially for the commercial potato at Cuba? And I saw something interesting. Maybe I should ask you. How do you do a budget of 216,000 for a study? How does that happen? Thanks.
Very much. So you may answer now. Very briefly, please.
Thank you for the questions. I like the interest that our presentation has generated in the minds of scientists and everybody here. To start with the first question asked by Dr. Peter, I didn't want to start with challenges with the beginning because it is so enormous that we may not carry it. The first, our only help is this project from Sahel. As Dr. Sumia asked, Sahel Consulting is the company that Sepia Foundation is passing through to reach us. And they fund them for the majority of the works we presented. In fact, up to 90% came from them. The challenges in handling this TPS work, the first one is screen house. We don't have any standard screen house at the station there in Jos. I thank God that with the expertise of the people available, we have been able to manage what is on ground to getting some results. And I appreciate Peter for his constant visits and persuasions to make the institute do well. apart from we have storage issues, but concentrating on TPS, we need a irrigation system, a proper irrigation system. We need a standard screen house with tables, and we need a standard medium that is used for this particular trial. I didn't want to bring in so many other things that may be bugging us, but particularly on this TPS, we need screen house and irrigation system. Every other thing can go, but without these two, we may not do much. Even though we have been trying with the little that is on ground, but it is really challenging. For Dr. Mrs. Anedu, I think there's a table that I showed that has the food composition, nutrient compositions of the Connect. We did not rush to release committee without having all the information on ground. The funders of the project, they are very much interested in getting all that is required for their purpose, for their own need, and for us. So the results is encompassing, but we don't have enough time to start presenting everything. Dr. Sawalam, thank you so much for this, your question. Please, it is a policy response that can address that. But what I know is beyond me. It's not what I can handle alone. But what I know is that when we adapt varieties from outside, and it happened that maybe during the course of evaluations, farmers pick it up, and due to the performance of the variety, they give it a particular name. It is usually easy for us to make that name to sell, but we still refer the question to the management so that they can give a proper response to that. In the title, as Dr. Asmia suggested, well taken, sir. For the resistance in a Connect potato for blight, there are levels of resistance. For now, in the world, not only in Nigeria, we have not had any potato that has complete resistance to lead blight. The only thing is that we have a high level of tolerance in this particular variety called Connect. It handles lead blight very well, including bacterial wilt. For the journey that we went to Plateau State with you in 2022, the measures that we recommended are being observed very well. The short-term measure is the one that is very well observed. For the medium measure, we have been able to release the 4 varieties, which you know, the Unica, the Baban, Kiao, and the Juria. And by the grace of God, we have been able to release this Connect now. So I will not want to attempt the question on GVPP. Dr. Madi is here. Please, I will want, sir, please, respond to the question, sir.
Okay, on the biotech potatoes, we are at the verge of submitting the dossier for deregulation to MBMA. So as soon as we do that, we hope that they will deregulate it, and then we'll proceed to do the remaining pre-release trials to have it officially released. So it's on course, though with a little delay because that project was upended by the change in administration because it was USAID funded.
Thank you, sir.
Thank you.
For
Thank you very much.
the one of Dr. Ma, sir, please, I will meet you later because that is a very source of information that can help us in future. Thank you so much.
Thank you very much. A round of applause for the potato program. Thank you. Like he said, of course, there's room for further dialogue and connection after now, whatever we still have in mind to ask. So next up is the Ginger program. Ginger program. from the program here, ginger program, you are given only 10 minutes. From the program, ginger program is given only 10 minutes. Okay, you have a total of 20 for everything, so please work within it. Thank you.
Thank you, Chairman, the ED, and every other person here. Welcome to the Ginger Pro. We are here to present our work in 2025 and our proposals for 2026. Next slide, please. Okay, one of our major achievements was the introduction of exotic plants, exotic germplasm from Morocco and Kenya to boost our genetic base, because so far majority of us know that what we have is homogene 1 and homogene 2, and this has affected our breeding line and also production. So with the introduction of these two exotic lines, we hope to increase production and breeding, and possibly, very possibly in a short time, be among release new varieties and be competing with cassava and yam. Also, we also try to develop some protocols to mitigate the cost of production in ginger. We have noticed that mostly mulch is a major problem in ginger production, and we are trying to see how we can mitigate it by pre-sprouting before we go to the field to eliminate the process of mulch in ginger. Next slide, please. Okay, under technology transfer, most of our staffs and scientists have been engaged in training and have served as resource personnel in training of farmers, and also we have engaged in farmers within the country in terms of resource personnel, and also providing technical support as regards to ginger production. And our need, like you stated there, we need an efficient irrigation system so that we can do all year-round production. And of course, you know, funding is research, and research is funding. Goals for 2026, increase our germplasm, which we have started, develop low-cost protocols, and disease resistance, develop disease resistance and control of insects and pests. Next slide, please. Okay, that's our staff strength. Is there next slide? Okay, for 2025, we have 5 approved work, and 3 were successful. 3 were approved, and the other 2, 1 completed and 2 ongoing. But I will talk a little about the 3 of them. Let's go, next slide. 2025 annual report. So like I said earlier, we introduced these 2 exotic plants from Morocco and ginger, S. Marrakesh and S. Togini, and 2 of them have been evaluated by breeders, and they showed promising traits. Like you can see on the screen, if you look at S. Marrakesh, if you look at the weight by plant, it's 3. And if you look at the weight by plant, it's 0.4, and it's more than the ones we have, the UG1 and the UG2. And down is the pictorial representation of those varieties. Next slide. So if you look at the morphological descriptors of those varieties, you can look at them. The S. Marrakesh and the S. Tigoni are pinkish, light pink, and light pink in color. The flesh color, you have medium yellow and light yellow as compared to our normal homogene 1 and homogene 2. So what are we trying to do next is to evaluate them for their traits, promising agronomical traits and nutritional quality, so that we can proceed with further work with them. Next slide. So this is an ongoing work, but I want to talk about it because I told you earlier what we are trying to do is to reduce the cost of production. So this is, we try to pre-sprout and compare it to those that we send mulch. So in the pre-sprouting, so we pre-sprouted before going to the field, and also we established some with mulch in the field. So the preliminary result is showing you that there's no significant difference in terms of yield for those that are pre-sprouted and sent to the field and those that are mulch. So though the yield varies, but significantly there's no difference. But like I said, this is a preliminary study which we need to carry out this year to validate our finding. And if it proves successful and good, then this can be a lead way of eliminating mulching in ginger production. Next slide, please. Okay. Next slide. Well, go a little back. Okay, next, the first one, next slide. So this also is part of our also trying to use cover crop as a mulch in terms of intercropping. So we try to see, okay, can we intercrop ginger with some leguminous crop like groundnut, soybean, cowpea, and to see the effect. In as much as the cover crop, these leguminous crops also we add nutrient to the soil. They can also serve as a mulch for the ginger. So we'll be hitting or mitigating the cost of mulch at the same time enriching the soil. So what we did here, next slide. So you look at it says sow ginger and groundnut. If you look at the picture, the sow ginger and groundnut ginger combination intercrop showed comparatively high yield, both in statistically similar to the plant, as you can see also in the slide. Next slide. Now, this is a regression analysis showing you the effect of temperature on the height and the yield of the plant. So as the temperature decreases, we observe that the yield decreases, likewise the height. Now, giving us that at about 26.5 to 27.5 degrees, that's a favorable temperature for ginger germination and sprouting. So this, like I said, there are preliminary studies. We need to repeat them this year, so to confirm our results so that we can categorically say, okay, farmers can intercrop ginger with other crop, eliminating mulching, or farmers can pre-sprout before going to the field so that they can also eliminate mulch and then increase production and yield. On that note, we come to our 2026 proposals. Now, the first proposal we have is the evaluation of the traits, quality traits for the newly exotic plants. So we are going to do some evaluation of these quality traits of this ginger and compare them with what we already have, the UG1 and UG2, to know how they perform. Already we know that in terms of yield, they are doing well, as we saw in the previous table. Next slide, please. Also, now, yesterday there was a panel of discussion here, what I said, why farmers low adoption, and it was that in our unavailability of the planting material. So while we are doing this, we want to use a single node technology to multiply the plants so that we can have more planting materials. That's the essence of this one. Then this one is on climate change. So we want to integrate the forest floor and also straw mulch to see how it affects the soil organic matter as regards to the production of ginger, the UG1 and the UG2 we have. Next slide. Okay, this is a continuation of the report we have. We're trying to look at pre-sprouting and also their performance in the field, and also comparing one single node and multiple nodes and to see the differences. The picture you see in that places are actually pictures of ginger coming from a single node. So, but this is just a preliminary study we did. Those ginger are coming from one single node, which we established in the screen house. Next slide, please. Okay, this is on plant health, and we are trying to look at using some fungicides to address some ginger diseases, pathogenic diseases. So, first, to identify the causative agents by doing some microbial studies, and then we use some fungicides to see whether they have an antifungal effect on the pathogens, and also to look at the quality of the ginger rhizomes after harvest. Next slide, please. Also, this is another proposal on plant health, trying to look at laboratory investigation of microbial interactions and suppression of ginger rhizome disease. So whether some interactions of some microbial organisms could be antagonistic to the ginger pathogens. This is just the essence of this work, to see some actions or how some microorganisms are, maybe their action may be antagonistic to the fungi affecting our ginger in the field. Next slide. Okay, this is extraction or characterization of ginger derived, nanoparticles. So we are trying to use some nanoparticles to address the issue of storability and stability of some ginger. So some nanoparticles will be extracted from the ginger rhizomes and see how they aid in the storability of ginger and also how they aid in the stability of the rhizomes. This is all this work is talking about. Next slide, please. Okay, this is our substation at Ibariam. They want to look at the effect of 3 organic manure in combination of NPK in the production and yield of ginger. Next slide, please. And the manure they want to look at poultry manure, look at pig dung and cow dung. Next slide also is from Ibariam. They want to look at the effect of NPK rates on the growth and yield of ginger. So looking at different levels of NPK on the growth and yield performance of ginger. Next slide, please. Impact of soil particles on the incidence and severity of ginger rhizomes root rot in Kaduna. Okay, this is from our substation in Kaduna. So they want to look at the impact. We were looking at our impact of soil particles on the incidence and severity of ginger rhizome disease. So one of the objectives, the last objective there is to identify soil management practices that could potentially mitigate rhizome root rot based on soil property analysis. Can I have the next slide, please? Analysis of farmer knowledge. This is coming from our socio-economists. So they want to look at the analysis of knowledge, attitude, and management strategies on the ginger disease outbreak on smallholder farmers. So they want to look at their perspective, what they do, how they have they heard, what are they doing, or what mitigation processes are they taking to address this issue of ginger disease. Next slide. This is also from our socioeconomists. So the socioeconomists in our team will be looking at the ginger blight outbreak, value chain disruption, stakeholder response in Nigeria ginger belt. So assess production and livelihood impacts, identify coping and adaptation strategies. So these are the proposals we have for 2026. And I say thank you, merci beaucoup.
Thank you very much. That's a wonderful presentation. Before we take any questions, I noticed this is a great leap from the number of experiments you had last year. So that is good. You had few last year, and this year you have increased the number and even the scope of the work in the program. Once more, please, can we give them a round of applause to the Ginger program? And thank you very much for staying within the time. So it's now time for questions. Let me look towards the side of the hall. Maybe I will see any hands up. So I'll ask with Dr. Anedu, Reverend, and then Professor Iwo Akpan, and then we come over to this side as usual. So Dr. Anedu, please. We'll start with Reverend Dr. Anedu. Over here. The light is right in my eyes. I can't see them.
Sorry.
Okay,
Yeah.
Dr. Henry will be after Professor Iwo before we come to this side. Thank you.
Yeah, I just want to ask a simple question. I think in 2014, there was a ginger blight that affected Nigeria's export of ginger. And then I think it was a national problem that the ministry had to give this institute a mandate to tackle that blight. How far have you gone with that? Are you able to arrest the situation?
Iwo is right behind. Excuse me, ma'am. Thank you.
Yes. Thank you. I want to start by making comments. This institute is the only institute in this country that has a mandate for ginger research. In 2008, they had over 10 accessions. In fact, almost 8 of them were exotic, from India, Mexico, and now you are talking about exotic varieties from Marrakesh, Morocco, and you have not mentioned anything about the old ones. 2009, carried out some work on the Nrcri G1 and UG2, and we're able to come out with 15 advanced lines, and it appears you've lost it. But I want to reassure you that we were able to recover some of those lines, and under the support of the TED Fund, we advanced them for multi-locational trials in 10 states of the Federation. But by the grace of God, by next month, we'll be ready. We will submit to TED Fund and also submit to this institute. And the issue of that is the blight. In isolation, because we don't have the mandate. Everything this institute, we will contact this institute and see how we can perform and evaluate those lines for their reaction to this blight at the place where they are endemic. So my comment now is, you people, the breeders in this unit, you people don't seem to be serious. You get materials and you allow them to get lost. A breeder, that's the major thing, is to maintain the lines no matter how old the materials are, because these are the germplasm where you can lay your hands on for further breeding. And there is a new innovation from the proposal you presented, single note trial, which is equivalent to a mini set in yam. In fact, if you succeed, that would be a breakthrough. And for those involved in ginger breeding, ginger is nothing but money now. Money. Ego. People look for material, planting materials. If you have a bucket of ginger, that is over 100,000 naira now. So people should be involved, and then also, if you can succeed in this year technology, that would be a major breakthrough. Thank you.
Thank you, Professor Iwo. So we go on to Dr. Harry, please. No, Dr. Harry, please. Dr. Harry, please. Thank you.
Standing on the existing protocol. The coordinator, we are happy from what you presented. First of all, I have only two comments. The first one is, after registration of ginger, two varieties, and the name was given as Umujim 1, Umujim 2. In accordance, Umujim 1, that's Umudike ginger. That is the local name. Professor Asawalam said. And you are still calling UG1, UG2. I don't think that should come after 2 or 3 years. It should be Umujim 1, Umujim 2. I don't know UG1 or whether you is the new one you brought from Morocco or Iran. That is one. Two, when we were here as a coordinator of ginger, there was something that came out in the field. Some people say it's fungi and all these things. And we were not able to establish that fungal disease. In fact, it was more of a moisture stress. And it comes towards the end, when the crop is finished, senescent. So, and it hasn't no significant yield difference. So we didn't want to bother ourselves to, continue, to do any research on that. For you now to have a proposal for fungal, something, it appears it's dead on arrival project. Because if you now say fungal disease and now recommend to farmers to apply fungicide, and they spend all their money, the fungicides are very costly, and then they start, it does not affect the yield. There are no significant yield difference. Then it's not a very good work to, continue. That's the two issues.
Thank you very much, Dr. Harris. So we can come to this side now. The same veterans. So just take your question very brief and go direct to the point. So we start with Dr. Muzo, we go to Dr. Ano, then we go right, and then we come to the front to Dr. Asomoga. Very briefly, please. Thank you.
Hello. Do I go ahead? Maybe everything I'm talking about has been sandwiched and plant health. But I want to say this. When you plant out in the field, the thing dies. Why? Many people will say soil disease. What is that soil disease? It's important to know that soil disease. When I was here, we tried to go through, I'm a nematologist. We tried to bring up a nematologist here. I don't know if nematode is being sidelined. Otherwise, Wadele, who is now at Jos, maybe that's why I saw the guy in Incognita. Because Wadele was here, we were trying to bring him up. But I see that within the institute, it's a management issue. Within the institute, I don't know if there's another nematologist here. Otherwise, the person that has been working on nematodes should come back and then be working through all the programs. There is no crop you plant here that you don't have nematodes attacking such. Instead, it's being said that it's something else. That's not right. So this is a management issue. I'm pleading with the ED and the team to see that there should be a nematologist who will be working around the programs. There's scarcely any crop you plant here, you don't have nematodes attacking it. Scarcely.
Thank you, sir. I'm sure the ED has heard. I wanted to call Professor Chilaka, and I made a mistake. That was the first name I wanted to call. Please come to Professor Chilaka before we go to the others. His hand is up. Thank you.
Is this on? Okay, thank you. Please, the moderator, please, let me correct the impression. What's his name? Oheri. UG1, I was a pioneer ginger researcher in this institute in the 70s. Now, UG1, UG2. UG means Umudike ginger. So what are you talking about? But if you want their local names, Tafingiwa and Yasumbiri. So my question is this. When people are the ginger program said they're intercropping, why did you choose groundnut to intercrop? Why didn't you go for, what was the choice? The need for groundnut. Why didn't you go for a goosey that we normally use along with all other root crops? And then your new introductions, how far do they compare in pungency when you put them side by side with your UG1 and UG2? Please.
Thank you, Prof. Prof echoed my thoughts because I was just asking one of the rapporteurs, why groundnut with ginger? I was just wondering. Thank you, sir. Yes. Dr. Ano, go ahead.
Thank you. My comment is just a brief one. Complementary use of organic manure and inorganic fertilizer. I don't know if you people have seen the work I did with late Dr. Emehute. It's published and is on this same area, complementary use of organic manure, various manures, and inorganic fertilizer on ginger, and is in the literature. Then you said extraction of nanoparticles from ginger. Nanoparticles, from the title, one would think you are going to use maybe a solvent or something, but it's a physical something. Maybe you want to reduce ginger to nanoparticles and now use it. Then the other one is soul ginger and ginger granite. You said soul ginger and ginger granite. You were comparing soul ginger and ginger granite systems. And you said soul ginger and ginger granite gave high yield. That's not comparison. If you had said soul ginger and ginger granite systems gave comparably and there was no significant difference between the yields, that will make some sense. Saying that all the two gave high yields, it doesn't convey any scientific opinion on comparison. Thank you.
Thank you, sir. Okay, please let him ask, and then finally we'll come to Dr. Asamoah, and that will be all for the questions for this session.
Yeah, mine is very brief. First and foremost, I want to congratulate the management for increasing the staff strength of Ginger program. Looking at what was presented, the number of research officers attached to that program has increased relative to what it used to be in the past. Time there was when it was only Dr. Echendo as the coordinator and myself alone. But today, the picture has changed, and I'm very happy about that. My observation from the presentation of Ginger program is that enough literature review was not properly done. We appear to be going round and round the same issue, maybe changing just one or two names and so on. All that they propose, apart from impact of soil properties on ginger rot, majority of what they propose have all been done. Unless there is any deficiency found in what was done, which now justifies a new approach. Otherwise, is it not a waste of resources repeating what has been done? I am very convinced that even the intercropping experiment is trying to evaluate has been done. And we found that a particular population of cowpea that can be intercropped with ginger to moderate the soil temperature and the soil moisture, which is essentially what mulching does in ginger cropping system. So I don't see the need as being valid, repeating all these things over and over again. Besides, granite that they have chosen, remember, you will need to, granite matures earlier than ginger for sure. And that will distort the configuration of the soil during harvest, which is likely going to affect the ginger rhizome. So I want to appeal that you look at that your proposal. If you must carry out more experiments on intercropping of ginger, let it be what has not been tried. Or if you find any deficiency in what was done in the past, you can come up with it, and then it will be done. I know we even extended this kind of trial under oil palm plantation environment and even rubber with Dr. Woods as the coordinator then. So I am suggesting, and strongly too, that we adopt the idea of reviewing literature exhaustively before we make our proposals in ginger program. Thank you very much.
Thank you, sir. Last but not the least, Dr. Asomugha.
Thank you so much, my co-host. These are the people who have worked on ginger, and they are telling you what to do and what has been done. So please, you have to go back and now Dr. Ewo raised the issue of exotic varieties we have in those days. Where are they? Maran, Himachal Pradesh, St. Vincent. These are varieties we have in our germplasm. Where are they now? Nobody is mentioning about them. Please, you have to tell us where they are. Where they are, yes, because the people suffer to bring things. Yes, and to maintain them. And you are telling us you don't know, you are going to Marrakesh and that and that. What has happened to our